don't look very enthusiastic today. Never. Did you get the email I sent yesterday? Okay, so just make sure you understand, my intention originally was to get through complement system and inflammation, and number one, we probably wouldn't have done that anyway. But number two, as I started making up the exam, there's a fair amount of material to cover on this exam. So what I'm going to do is I'm going to go through just a little bit of additional stuff today, and I'll tell you where that break point is. Then if there are no questions, I'll continue on with complement and inflammation. But those will be on the final exam, not this exam. So the study guides that I sent you have been adjusted to reflect that breakdown of information. I haven't really changed, so if you've done study guide questions, I haven't changed the study guide questions. But on the final study guide, which was line 1 and line 2, I took some stuff off of there and moved it over to the final exam. Okay? So use those to study for your exam. So, questions? Okay, we spent some time last period talking about a very major mechanism of the immune system killing pathogenic cells, and that is phagocytosis. We talked about the steps in phagocytosis, ultimately getting inside of that phagolysosome and being exposed to all those reactive oxygen species and all those hydrolytic enzymes results in destruction of the pathogen. Okay? What I want to talk about today a little bit is some non-phagocytic mechanisms that cells can use to kill pathogens as well. We've already mentioned one of them briefly. I'll actually mention two of them. Natural killer cells and the T cytotoxic or killer cells. Remember that these are both agranulocytic cells, and in both cases what these immune cells are doing is they're basically secreting molecules onto the surface of the pathogen and punching holes in them, and then other enzymes from the immune cells can enter those cells then through those holes and cause those cells to die through apoptosis. And we're going to come back and talk about the T killer cells a little bit more and get into a little bit more detail about that perforin granzyme process that results in cell death. But are there some granulocytic mechanisms other than phagocytosis? And the answer is yeah. Neutrophils, which are a primary phagocytic cell, have some additional mechanisms to kill pathogens that don't involve phagocytosis. So, for example, on the neutrophil, if the TLR binds a PAM, okay, what's TLR? Toll-like receptor. Okay, that's the receptor on the surface of the neutrophil. And it is looking for these pathogen-associated molecular patterns, these PAMs. Okay? And if the two bind, then that can activate the neutrophil to do a couple of additional things to kill pathogens besides engulf it and chew it up. Okay? One of the things that can happen is that binding of that toll-like receptor to the pathogen can cause that neutrophil to start secreting hypochlorite. Remember hypochlorite? Okay, that's one of the halogen compounds, and that is one of the ways in which we can control the growth of microorganisms through chemical control. Another thing that these cells can start doing is binding of that toll-like receptor to the PAM can activate membrane proteins, enzymes, which result in the production of nitric oxide. This is not the same as nitrous oxide. Some of you might have heard of that before. Maybe even had it at the dentist, the laughing gas. Okay. But nitric oxide is a very powerful stimulator of inflammation. And in a few minutes, if we have time, we'll talk about inflammation and how inflammation can result in the elimination of pathogens as well. And then finally, even in their death, these neutrophils can produce these interesting structures called neutrophil nets. And basically what happens is when these cells lyse, they release their DNA and their protein into the environment. And the DNA and the protein starts complexing with each other and forming this extracellular matrix that really does look like a net. You can see it on this slide right up here. So this Klebsiella cell is in the process of lysing and releasing its DNA and protein and forming this external net, which can kind of help to trap pathogens. That's a similar phenomenon to what occurs in inflammation. One of the things that's stimulated in inflammation is blood clotting. And remember, clotting is another way in which the immune system can try to localize and trap a pathogen and keeping it from spreading. What adaptation have some bacteria evolved to break out of a clot? Put it on by Thursday. Exoenzymes like streptokinase can bust those clots. Okay. Will bacteria eventually evolve some way to break up this net? Probably. Probably. The relationship between pathogen and the immune system is a constantly evolving situation. And when one system evolves, the other counter evolves. So I would not be surprised to see bacteria evolve some mechanism to bust out of these nets that are produced. When we talked about granulocytic cells, we briefly mentioned the eosinophils. And these are the granulocytic cells that provide most of our immunity against parasitic worms, like tapeworms, for example. And these particular eosinophils can kill tapeworms by secreting chemicals onto their surface, which have cell cytotoxicity. So it actually can just attach to these particular tapeworm cells on their surface and start destroying those cells. So phagocytosis isn't the only way that these granulocytic cells can kill. There are other sophisticated mechanisms as well. And then finally, there's kind of a general mechanism that potentially all cells can use to kill and inactivate virus. These chemicals that are secreted by most cells of the body, there are certain cells that produce more of these than others. White blood cells and fibroblasts, which are associated with connective tissue, are particularly adept at producing these interferons. But epithelial cells can produce them. It's kind of a generalized defense mechanism against viral infection. Have you ever heard of interferons before? They literally interfere with viral replication. Now, what's interesting about these particular substances is they don't protect the cell that produces them. So if a virus infects a cell and it starts producing interferon, That's not going to help that cell that's been infected. But the interferon then can be released from the infected cell and diffuse out and bind to uninfected cells in the neighborhood. And the uninfected cells can then produce antiviral proteins if they become infected. So the cell that produces the interferon is a goner. But in its death, it produces chemical compounds that get cells ready, should they be infected, to fight off that infection. In reality, there are actually two types of interferon. And they are just designated type 1 and type 2. Type 1 interferons are the ones that are responsible for these antiviral proteins being produced. And actually, there are two classes of type 1 interferons called alpha and beta. They do the same thing. They're just produced by different types of cells, and I'll show you a little table in a second that shows you typically which type of cells produce type 1 alpha and which ones produce type beta, type 1 beta. Type 2 work differently. Type 2 interferons have a totally different function. They are immune stimulants. They tend to work a little bit later in the infection process, and they can stimulate phagocytosis, for example, by neutrophils and macrophages. They can help to keep macrophages at the site of infection so they don't wander away. There's a type of type 2 interferon called macrophage inhibition factor. And so they really play a different role of kind of assisting other aspects of the immune system and juicing them up to be a little bit better at doing the job they normally would do anyway. So here's our type 1 alpha and beta interferon. And here is a virus that has affected a cell. Okay? That virus starts to replicate, and that viral replication starts to trigger transcription of the normally dormant interferon gene. And it starts producing the interferon as it's dying. And eventually this cell will die in lysis. Okay? And when it dies in lysis, it will release new virions, which could affect new cells. But that interferon diffuses out into the environment and can attach to receptors on the surface of healthy cells. And when that happens, that activates a gene in the genome for antiviral proteins. So now if that cell is infected by a virus, those antiviral proteins selectively start inhibiting nucleic acid synthesis of the virus. And it chops up the virus really before it can replicate. So in some respects, that's kind of like what a restriction enzyme is. Remember restriction enzymes, which we use in genetic engineering, chop up DNA, are adaptations by bacterial cells to resist viral infection. So in some respects, these antiviral proteins are kind of analogous to those restriction enzymes. There was a lot of excitement about interferon when it was first discovered, particularly the immune regulation aspect of interferon. And actually, interferon is still used in some treatments. For example, it is used routinely in treatment of multiple sclerosis. But for a while, people got so excited and they thought, wow, this drug is really going to help us cure cancer. Well, there were some limitations to interferon. It's a protein. It's pretty small molecular weight. It's very difficult to isolate. And cells don't produce much of it, tiny amounts. So to do the first therapeutic trials, particularly in cancer treatment with interferon, it was just god-awfully expensive. Because what they had to do was they had to raise up cells in tissue culture, grow some fibroblasts, for example, and then lyse those cells and isolate the interferon. And it cost hundreds of thousands of dollars to be able to get enough to do just a trial on one person. So it was really pretty cost prohibitive. Eventually, they were able to identify and clone the human interferon gene and insert that into bacteria. And so now interferon can be produced by recombinant DNA technology the same way that insulin can be produced. So the cost has really gone down quite a bit. It's fairly short-lived, and so you have to keep applying it. There has been ongoing research trying to use interferon delivered in a nasal spray to try to prevent colds from occurring. Okay? Prime those cells of the nasal cavity with interferon so that they will produce antiviral proteins, and then if they're infected by the cold virus, they will resist it. Haven't heard any update on that recently. It's been in clinical trials for quite a while, I believe. Another thing that is difficult about interferon is that it is highly species specific. So mouse interferon, for example, will not prime human cells to produce antiviral protein. So if we really want to have therapeutic interferon, it's got to come from the human genome. Now that we've cloned the gene, that's not that big of a problem.
Does that mean like human testing would have to be done like before any testing is done in animals? Like they could use the...
I think that they could do animal testing with animal interferon on animals.
Okay.
Okay.
And they would assume to have the same effects.
Yeah, yeah. And then when you move to the human testing, you'd obviously have to
Yeah.
switch over and use the human interferon. Yep.
I don't know if this is a silly question, but like using just even with like the nasal spray, wouldn't that kind of go back to the treatment like superbugs and things like that? If that would be used, or is it since it's like a protein that we produce that doesn't play a role?
Well, yeah, it definitely is a protein we produce. We normally only produce it
Right.
in response to viral infection. So, since it activates a gene, could you become resistant to it? That's a good question. Because I guess if more antiviral proteins are being produced, and could microorganisms evolve a way to resist those antiviral proteins? I suppose that's, yeah, that's probably a legitimate concern. So here are the kind of breakdowns of differences between type 1 and type 2 interferon. Type 1, so the alpha and beta. Again, both stimulate production of antiviral proteins. Really the big differences is alphas are produced primarily by epithelial cells, and a lot of white blood cells produce these interferons. And beta are produced more by the fibroblasts and connective cell tissues. In fact, beta interferons used to be called fibroblast interferon, and alpha interferons used to be called leukocyte interferon. This is what gets really, really frustrating sometimes about all of these chemical cytokines, is that different people discovered them in different labs, and when they discovered them, they gave them names. And there's not complete standardization of the names yet. So many of the same cytokines have multiple synonyms for names. And I think that there's a real effort to try to standardize some of that stuff. But there's still a lot of this. Interleukin 10 is an macrophage inhibition factor. It really makes it much more confusing than it should be. Type 2s, which are also called gamma interferons, are produced by both T lymphocytes and the natural killer cells. And they tend to stimulate and enhance phagocytosis by both macrophages and neutrophils. Okay, put a big line in your notes. And this is where we will stop for the exam. So before I forge on, questions? Or just save them all for tomorrow? Yes, sir.
So is diabetes independent of histamine reactions? Because I know histamines increase the permeability of the capillaries.
I'm sorry, it's what?
Diabetes or
Diapetesis.
diabetes.
Diapetesis is enhanced by inflammation because if those endothelial cells have very tight junctions, it's really difficult for those white blood cells to crawl out of those vessels. But if we loosen those junctions up as the vessels dilate, then that enhances the rate of diapetesis. Okay.
Are we recording?
Yes. We are recording.
Start with chemotherapy.
Yes.
We'll start with chemotherapy. Yep. So chemotherapy, we started out talking about, some of the history, the sulfonamides, spent most of our time on the antibiotics. Then we jumped into the kind of host-parasite relationship, some of the characteristics of pathogens, the exoenzymes, the toxins, etc. Ended up with epidemiology. And then this section covers the first line of immunity, the barriers, the chemicals. And then the second line got us into the blood and lymphatic compartments with all the blood cells and stuff. Yeah, there's a fair amount of stuff. And so I think that cutting it off now and not including complement system and inflammation is probably a good idea. Of course, it will be on the final exam, but
I'll worry about that.
you'll worry about that then.
What's transient flora?
Transient flora is flora that you acquire and develop for just a short period of time, and then it is either removed physically as the skin sheds, or it's outcompeted by that normal flora. But sometimes in a weakened state of immunity, transients can cause infection.
And that's associated with the germ-free animals, like we take some of their flora for a little?
The reason that we talked about germ-free animals was the study of germ-free animals, or azentic animals, really helped us to appreciate the role that normal flora plays in a number of facets of an animal's life. So there was a chart we gave, and it showed kind of comparison and contrasting germ-free versus non-germ-free animals, and kind of what the differences were. Just curious, did anybody ever look at that website I sent you with the access to the study materials and all that stuff for? It's not a book we use, but it's a book I use a lot and kind of prepping for.
Are you suggesting?
I mean, there's some good stuff on there. There's some good animations on there. There are, practice questions. I was just curious if anybody had taken advantage of it.
Well, I figured out how to find that patient zero illustrated by the chronicle.
Oh, yeah?
So they found out, when they eliminated it because they found out that it was like all over the country, so it was somebody who was well traveled. And then they isolated it then to, like different, heterosexual bars. And then they found out it was Canadian Air. By isolating it, then it was like Canadian Air pilots. Or like a
Wait, this was for HIV or Ebola?
This was for HIV.
Okay.
He said like it's a lot harder, especially if it could be transmitted through the air.
Yeah.
He said there's nothing really that is
Just we thought they were harmless.
Well, again, there will be some time tomorrow because, like I say, really all we have to do is we have to inoculate our tubes, and then we'll do a little exercise to kind of get you thinking about how to do a flow diagram, because you are going to have to put together a flow diagram for some organisms, and then I'm going to give you a little unknown, and you're going to use your flow diagram and some media to tell me what they are.
Do we need to know the difference between structures and antibiotics? So back to like the R groups that we
No, I mean, I threw the pictures up there so you could see kind of the diversity of structures of different classes of antibiotics. I do think it's important to know that penicillins and cephalosporins, for example, are beta-lactam antibiotics and have that beta-lactam nucleus. But in terms of what the specific R groups are, or even, I suppose, tetracycline is one that you could recognize fairly early because it has 4 of those heterocyclic rings. It looks very different from the beta-lactam antibiotics, but I never make you memorize structures. It's not something I do.
Do we need to know any specific bacteria species, names, and whatnot?
Any specific bacteria species?
Because
If we talked about them in the context of a particular disease or a particular toxin, then yeah, you should. Now, there's not that many. I mean, many of them are Clostridium species. So, Clostridium
My
botulinum.
question's too vague.
Pardon me?
Maybe my question's too vague.
Okay.
There is a lot.
I'm trying to think.
It's
What,
like, I don't mean like, just like, if you have to study guy like Staphylococcus, like, that's different. So like, you have like all these enzymes that like examples of diseases and then species within those, or you would just like know the disease.
Yeah, I mean, I do think that you should know, for example, the major Clostridium species that cause those particular diseases. There's botulinum that causes botulism. There's tetani, which causes tetanus, and there's the Perfringes, which causes gas gangrene. Other than that, I talked about staph and strep kind of as a genus, but do you have to know which species of staph cause particular diseases like impetigo or scalded skin syndrome or something like that? No, you do not. Okay.
In the textbook, it talks a lot about, or let you know what he did and how he did it, but it never really mentioned the other, the HABA guy. Do we need to know his contribution?
He was just a co-worker with
Was there-
Airly. Yeah.
Okay, because
Yep.
it never said his name in the textbooks.
Yep. And, again, I mean, use what I put in the lectures and the lecture notes as kind of a guide. I mean, so I mentioned Ehrlich in the context of this was a guy, and he worked with HABA, that was trying to develop the magic bullet. And the magic bullet was a selective drug that would kill the pathogen and not the host. And he really never found it. The salvarsan that he developed was not a very magic bullet because it really was pretty nasty to the host as well. So use the lecture's notes as kind of a guide for how much detail and depth you need to go into. The book goes into more depth on some things and less depth on other things. So I consider the book more as kind of a reference to set the context. And then if we do go into some detail, the book can help to reinforce that. But if the book does something in detail and I don't, no, you don't have to know that. Yes.
How is the Clostridium perfringens adapted to promote gangrene?
How is it adapted to promote gangrene? Primarily because of the exoenzymes that it produces, which allows it to spread. And as it spreads, it kills new tissue. And as that new tissue then necroses, that pocket becomes anaerobic. So then the bacteria can kind of move into that new anaerobic environment. So it's kind of like they send stuff out to kill, and then they move it and inhabit that killed area.
And then they just keep going.
Yep,
and going.
yep. Okay.
Can you go over wet and dry again?
Wet and dry? Okay. Dry gangrene results from ischemia, blood flow. And that could be because of a crushing injury, for example. It could also be the result of hypothermia, frostbite, where the blood vessels constrict to try to preserve body core heat. But when the blood flow becomes blocked for some reason or another, cells start to die. And then tissue starts to necrose. And lysosomes break open. And then they flood out and start attacking healthy tissue. And then those cells die and they necrose. Okay? So there's not a bacterial involvement in dry gangrene. It's all the result of ischemia. Okay? In wet gangrene, there's bacterial involvement. And the blocked blood flow could result from trauma, but then as the bacterial spores that get into that trauma site start producing all their nasty enzymes, and gas starts to be produced and stuff, that swelling can further block blood flow. They start, digesting the muscle proteins, producing the lecithinase, and then basically now that was what we're talking to Nicole about, is they start then sending out these chemicals which kill adjacent cells, and then they can move into that environment.
But the wet gangrene can also be initiated like by ischemia as well, right? Or is that-
But there's got to be,
But it's because of the bacteria.
yes.
So it can happen without this.
That's
Dry.
dry. Okay.
Yeah, in both cases there's ischemia. There's blocked blood flow.
Okay.
One's with bacteria, one's without.
One's pseudobacterial, one's not. Okay. You look
Yeah,
confused
that
then.
makes- no, that makes sense.
Okay.
So the bacteria doesn't cause the lack of blood flow.
It contributes to the lack of blood flow.
Well, but it doesn't initially start. Like, it's-
No, probably the trauma would. Yep. Now, sometimes situations like in diabetes, the lack of blood flow that causes tissue necrosis, and then we get a co-infection with the bacteria. Okay. Yes.
I just need to refresh my memory on the drug resistance involving mutation R factors. What were the 4 examples for the drug inactivity?
Well, okay, the first thing we talked about in regard to drug resistance was that it really is kind of a genetic phenomena for a couple of reasons. Number one is that drug resistance results as a result of mutation, ultimately. Okay? And those mutations can then result in enzymes like beta-lactamases that can inactivate the drug, so that was one mechanism. They could result in active drug pumps, which pump the drugs out of the cytoplasm. They could result in the change of receptors on the surface of ribosomes, for example, so the antibiotics can no longer bind. I'm missing one. There was at least another one.
There's a whole bunch.
Decreased permeability.
Then there's also like all 2 different metabolic pathways.
Yeah, metabolic shunt. Okay, so that the
So there's more than 4, right?
Yeah.
Oh, okay.
yeah, typically when I ask for a number, I've given you a list that has more than that number, so you've got some latitude to pick and choose. Okay. The other genetic aspect of, what's the other genetic aspect of drug resistance? R factors, okay? So once one individual has developed that mutation can spread because oftentimes that is a gene encoded on a plasmid, and the plasmid can be transferred by conjugation. We call those R factors or R plasmids if they have a resistance gene on them. Okay, Audrey, you have your hand up.
Yeah, I know that like pleiotropic is like talk about sickle cell and malaria. Can you go
We were just talking about genetic factors that can influence whether you are susceptible to a disease or not. And one of the genetic factors we talked about was if you don't have the proper receptors for a virus, it will infect you. So that's why there are oftentimes species barriers between infection. But then we also mentioned pleiotropic genes, like the sickle cell gene. A pleiotropic gene is a gene that influences more than one trait. So,
I like the calico cats.
calico cats are another.
That's a different thing.
It's really overactive.
It's been so long since I've had genetics that I can't remember what category that falls into. It's not the same. It's not pleiotropic.
Okay.
But anyway, what happens is that particular gene influences both sickle cell disease and malarial resistance. And basically the deal is that if you're heterozygote, then you have a resistance to malaria, but you are only a carrier of sickle cell disease. So that's really the only context that I tested.
Can we talk about the virulence factors of Staphylococcus?
The virulence factors of Staphylococcus. Yeah.
That's
What do
a
you-
long one.
Yeah. And there again, I think that if I ask you something like that, I'm going to ask you to give me a few examples as opposed to the whole exhaustive
Okay.
list.
Okay.
Okay? I mean, some of the major ones that just come to mind are capsules, for example. Okay? Oh, virulence factors, sorry. Not anti-phagocytic factors. Okay? Yeah, virulence factors. Am I going to tell you which ones you have to know? Something like that would be, if I asked you, would be more part of an open-ended question where you could call on maybe a couple of those as an example.
Like a short essay?
Yeah,
Okay.
yeah. I'm not going to give you an essay question and say, or a multiple choice question and say, which of these are virulence factors of Staphylococcus aureus?
Okay.
Okay. Yes.
There's a question that says, what are nosocomial infections? How do you say that?
That is the synonym for hospital-acquired infections, the HAI. We used to call them nosocomial, infections, and now the new verbiage is HAI, hospital-acquired infections.
Okay.
Kind of good, your hand.
Yeah, there was a question about metagenomics, and in my notes I just had that you just put like genetic fingerprints. Is that all that we really need to know about?
Yeah, I was looking back through the review questions, and I don't change those every semester. And I think last year I got a little bit more into metagenomics and this whole idea of you can identify organisms by genetic analysis in the presence of unique RNA sequences, because a lot of times it's really difficult to grow them and identify them. And that's kind of the strategy that's been used. I think we talked about that in the context of the Human Microbiome Project, and that they used that strategy to try to figure out what you had in your guts in various places as opposed to trying to culture all these things. So yeah, it's a strategy that uses particularly unique RNA sequences to infer the existence of microbial species, which are not actually growing and identifying. Really, it's like a foreign language in many respects, isn't it? There's a lot of A lot of vocabulary.
The common reserve for a human body, would that be like the mouth? I feel like that's just bacteria.
Well, when we talk about reservoirs, we talk about typically not a specific location, but whether a human is the whole reservoir where kind of it resides, or whether it's an animal, or whether it's soil or water.
Okay.
Okay, so typically I don't break it down and say a specific location. Now, each different part of the body we talked about has kind of unique normal flora. Okay, so there's all kinds of little microhabitats all around your body that have different normal flora depending upon their temperature, their pH. Okay. Yes.
Can you brush me up on ways we can minimize bacterial systems? Because like I keep
Ways we
confusing
can-
it with like what we can do to help treat it.
Well, the ways we can minimize antimicrobial resistance fall into some major categories, which then kind of have subcategories of more explanation. But there's two major strategies you can use, and one is limit the use of antibiotics. Okay? So don't put them in animal feed is one way to limit the use of antibiotics. Okay? Don't prescribe them for viral infections because they don't work. Okay? Don't use them if an individual is resistant to that antibiotic. And that's where we then got into some, okay, how do you know whether there is resistance? Okay, so you can do the Kirby-Bauer test, the Auger diffusion one. You can do the E-test. You can do the minimum inhibitory concentration test. So we kind of built on that a little bit. Don't use broad spectrum antibiotics. Try to use narrow spectrum antibiotics. Okay, so targeted antibiotic therapy. Take your antibiotics throughout the course of treatment. Don't discontinue use. Possibly combine antibiotics. Because the real fear is that if cells survive, they are going to be the resistant ones, and then they will give rise to populations which are now resistant. So if we're going to use antibiotics, we want to make sure we do it in such a way that they are effective and that they kill all of those cells so there are none surviving to reestablish a population. Does that help?
Yeah, it's just, it is very overlapping with describing the 4 factors that contribute to their alarming rate of development. So it's like trying to separate the two is
Mhm.
kind of impossible.
Yeah. Audrey?
I feel like they usually give like 2 antibiotics, like someone comes from that, and their infection is gone. Would it be better, would it like help resistance if they would give them like 2 antibiotics like initially?
Initially, that's my understanding. And it doesn't always have to even be an antibiotic. Have you ever heard of Augmentin? Did I talk about Augmentin? It's in the notes. It's amoxicillin plus a chemical called clavulanic acid. And clavulanic acid inhibits the beta-lactamase enzyme, which can degrade penicillin. And so this chemical augments the action of this amoxicillin, penicillin. And so that's a situation where they purposely combine 2 things and then initially give that to you.
Can you remind me what that E-test is again?
The E-test is a way to test for susceptibility quantitatively. It gives you a minimum inhibitory concentration, and you can do that through a series of diluted broths, for example, or it's got that stick
Okay.
you can stick on the auger,
Okay.
and you get that weird kind of oval-shaped inhibition, and you can read where the inhibition stops off the strip because it's got a gradation of antibiotics on it. So that's the E-test.
And that helps you determine the therapeutic.
Yes, minimum inhibition concentration is needed to calculate
Okay.
therapeutic index. Yes.
So protein A just prevents antibodies from binding to the pathogen, correct?
Yes. Fun, huh? Well, like I say, if you have more questions tomorrow, there will be time. And then I guess,
Would you highly suggest us to read the lab, like the last 2 weeks?
I highly suggest you read that.
Highly suggest.
There is probably less information in this lab that knowing ahead of time would be really helpful for the lab. In fact, in this lab, much of the information in the lab handout is more applicable on Friday when we interpret results.
So Friday.
You're angling for some bonus points, huh?
Always.
Always,
I'm always wheeling and dealing.
yes.
Can you read the anti?
Hey, I don't hear you guys complaining about
Put
it.
Altitudes right now.
Are these the same as the one, like the leukocytes, leukocytes?
Oh, we were definitely laughing over here.
The main anaphagocytic factors are the capsule and mechanisms that allow pathogens to survive inside of lysosomes. And I didn't tell you how those work.
Do they survive in the lysosome, or do they prevent the lysosome from binding to the phagosome?
Both. Yep.
Okay, so for number 16, you say give 3 different examples, or just like take those when you already know it's fine actual meals. Okay. We already answered. So I can talk about like what has collagenase or
Well, those are virulence factors. They're not antiphagocytic.
specifically what
Virulence factors are things that help invasion. And they're things like the exoenzymes.
the staphylococcus.
Yes.
They laid the dollar.
It's a toxin enzyme. Got it.
Okay?
Yeah.
Yeah.
We'll be back with more questions.
Okay.
We'll have
We'll be back with your numbers.
I get out at 5:30, but it's probably going to be closer to.
I wanted to make sure you were fully confident that was the one you.
I don't need to